In this video, looking at the estimation of pentose sugars in a given unknown sample. So the principle is acid hydrolysis of DNI releases ribose in the presence of strong acid dehydrates to eat furfural. Then orcinol further reacts with furfural to form a bluish green color complex, which is spectrometrically estimated. The required materials are ribose, distilled water, orcinol reagent, beaker, test tubes, cuvette, glass rod, SMF. Required materials pipettes, test tube stand, colorimeter, and boiling water bath. We're looking at the illustration and the reagent preparation in the illustration. So meanwhile, we are looking at what are pentoses. Pentoses is a monosaccharide, simple sugar with 5 carbon atoms. The chemical formula of all pentoses is C5H10O5, and the molecular weight is around 150.13 gram per mole. So pentoses are highly important and essential in the biochemistry. Ribose is a constituent of RNA, and the related molecule, deoxyribose, is a constituent of DNI. Phosphorylated pentoses are important products of the pentose phosphate pathway. Most importantly, ribose 5-phosphate, which is used in the synthesis of nucleotides and nucleic acids, and erythrose 4-phosphate, which is used in the synthesis of aromatic amino acids. Like some other monosaccharides, pentose exists in 2 forms, which is the open chain, linear form, or a closed chain cyclic form that easily convert into each other in water solutions. The linear form of pentoses, which usually exists in solutions, has an open chain backbone of 5 carbons. 4 of these carbons have 1 hydroxyl group, each connected by a single bond, and 1 has an oxygen atom connected by a double bond, forming a carbonyl group. The remaining bonds of the carbon atoms satisfied by 6 hydrogen atoms. That's the structure of the linear form is. So when you take a look at the classification of acid pentoses, subclass of pentoses, which is in the linear form, have the carbonyl carbon at carbon 1, forming an aldehyde derivative with structure. The most important example is ribose. The ketopentoses, instead of having the carbonyl at position 2 and 3, forming a ketone derivative. So when you take as all the pentoses have 3 chiral centers, therefore 8 different stereoisomers are possible. Ketopentoses have 2 chiral centers, therefore 4 different stereoisomers are possible. The 3 ketopentoses are in fact rare. So the properties in cell pentoses have a higher metabolic stability than hexoses. So a polymer composed of pentose sugars is called a pentosane. So you're looking at, when you take into consideration how to estimate pentoses, it is how what you're looking at here in this illustration, converting the pentose into furfural, which then reacts with the chromophore in Tollens test for pentoses. The furfural ring reacts with to form a colored compound. So in the illustration has 5 samples, 2 test solutions, and a blank. The volume of the different working standards, concentration of the working standard, and they are made of distilled water and the addition of orcinol reagent and incubation in a boiling water bath. So the quantitative estimation of pentose here, the pentoses are heated with concentrated self HCl. Furfural is formed, which contains it with orcinol. The presence of ferric ions, Fe3+, to give a bluish green color. So can be estimated at body the optical density of around 665 nanometer. So orcinol reagent is prepared in concentrated sulfuric acid, ribose. So you can see the addition of orcinol reagent in the mixture in the illustration. So 3 ml of orcinol reagent is added in each and every test tube. So in the illustration, orcinol is being added in each and every test tube after the addition of orcinol, the mixture. Each test tubes are kept in a boiling water bath, water at its boiling point, in a boiling water bath incubator for 25 to 30 minutes. You can see the bluish color developed in the illustration. So in the colorimeter or spectrophotometer, the optical density is set at 665 nanometer, and the bluish colored complex formed in each and every test tube from the test solutions, from the samples, from the blank or read out at 665 nanometer. You can see blank S1, S2, S3, S4, S5, and the test solution T1. So when you say the absorbance of unknown by the concentration of unknown is equal to absorbance of standard by concentration of standard. This is how you calculate the concentration of unknown solution. So the estimation of pentoses is this method is an example of condensation reaction, also an example of condensation reaction. Now you can see in the illustration so you can see the tabulation test working standard, the ODI at 665 nanometer. You can see the graph, concentration of working standard, microgram, plotted against the optical density. You can see the slope. So 0.2 ml of unknown solution contain 40 microgram, so 100 ml of unknown solution contain 20 microgram. So the amount of present stress